thank you. It's a pleasure to be here. And Lynn has done a great job already. She explained half of my talk, so I think I'm done. We won't take long. No, absolutely, yes. But nevertheless, let me go to the beginnings and try to explain this from the start. So we all need to eat. So there's a lot of this idea that we need to consume right amount of proteins, nutrients in general. They're not being consumed and then used immediately. They need to be sensed. There is a lot of machinery in a cell that tries to measure the amount of proteins, the amount of growth factors, lipids and carbohydrates, energy, and all of this needs to be orchestrated into a specific signal, an mTOR complex 1. So mTOR, as we know it, is something that is one of these hubs that so one of these hubs that tells the cell we have enough of everything, we can go on and build new proteins and grow our cells and develop our bodies. At the same time, it switches off catabolic processes and it prevents degradation. But of course, all of us, and it's not just us, it's everyone from a yeast to a human, need to survive in periods when we don't have enough nutrients. And this is particularly important for when we were in the wild, obviously, but even now, all these cultural practices of, for example, fasting, or if we choose to do dietary restriction, we need our bodies need to survive in these periods. And this is when these cascades that actually tell the cell that nutrients are available switch off, and instead another pathway activates. And so again, there's a whole series of phosphorylation events, protein kinase OLK1, VPS34, but all of this leads to the formation of this quite enigmatic membranes inside the cell. It's almost very difficult to place exactly where it arises, and there's a lot of research trying to understand what it is, but it's like a bubble inside the cell that flattens like a flat balloon, and then starts engulfing something inside the cell with an idea to wrap it up, and it could be anything, could be portions of cytoplasm, it could be organelles and lipid droplets, etc., to seal them up and then fuse with the lysosome. Lysosome is an organelle that allows for degradation of things. It's a bag full of proteases and other enzymatic enzymes, and all of this allows for the release of those building blocks back to the cell. So amino acids, fatty acids, etc. are released and refeed the system from within. So we only survive during periods of starvation because our cell is refeeding us from inside, and it reactivates the same nutrient sensing pathway. So as a result, we have this nice rebalance of the system. We start maintaining our cell growth, etc. And so it is about life and death. In this case, it's about being able to survive when we don't have nutrients. My interest in this field has been over 15 years, since I was a postdoc in David Rubinstein's lab in Cambridge, and since starting the lab in 2011 in Newcastle, and we've been working on these pathways and intricate relationships for quite some time. One thing is I'm focusing on today is not just about life and death, it's about living well, it's about health. Because that degradative cascade that I was just describing is not just providing nutrients, it's also allowing for the cell to ensure quality control to everything inside the cell. And autophagy degrades specific damaging agents inside the cell. It could be protein aggregates, it could be damaged organelles, it prevents DNA damage accumulation, even intracellular pathogens. The issue with this is that autophagy goes down with age, as we know it, and so this gradual decline in a quality control that is lost with time leads to this systemic effect, that what we essentially know as aging, systemic gradual decline in our physiology, increased inflammation, and those diseases that are typically associated with aging are essentially a consequence of this. So autophagy and its decline is something that is one of these underlying mechanisms that result in aging. And in the lab, we're interested in understanding how autophagy is able to pick up those damaged components of the cell. Specifically, how does it target them? What are the cellular functions of autophagy and what is the importance of it, essentially? And ultimately, as a sort of more practical application, is how can we target it to ensure longer and healthier life? Now, so the selectivity within autophagy is typically orchestrated by receptors. So this looks like a gobbledygook here. It's one protein. It's an example of a receptor. All you need to know here is that the function of a receptor is twofold. One is to bind to a ubiquitinated cargo. It could be anything in a cell. Typically, anything that is damaged would be ubiquitinated, and therefore is able to recognize that. The other part recognizes the nascent autophagic membrane. So it can bring that cargo to the autophagosome for degradation. As it does, it commits suicide at the same time because it gets degraded together with the cargo. Another important element of receptors is that they like to be oligomerized. So they need to clump together because autophagy likes bulkier cargo, so it needs to degrade something larger. And different receptors are doing different jobs in the cell. So, for example, P62 has been ascribed a role in degradation of protein aggregates. For example, other receptors would be playing a role in degradation of other intracellular components. And this list is definitely not exhaustive. This is very simplistic diagram. But what this means is that this multiple process of selective degradation of specific cargo is essentially what we know as being autophagy, and this is what we think is important for normal function of the cell. We sort of started and developed this concept that the signal that comes from these damaged intracellular components is actually mediated by reactive oxygen species. So we think, and that's almost like a spoiler for the rest of the talk, is that these reactive oxygen species oxidize these receptors and trigger their oligomerization, and this is what tells the cell to degrade those components. So not only autophagy in this case maintains protein and organellar homeostasis, it also maintains redox homeostasis, because by doing this, it also prevents intracellular components that are actually producing that increased levels of reactive oxygen species and therefore leading to oxidative stress. This has started quite some time ago, essentially when I started in Newcastle, and these three people, I like usually putting people in a lab that actually did the job because I do nothing nowadays. So Bernie Carroll, Gisela Otten, and Diego Mani, they spend a lot of time trying to basically understand how this process regulates P62, this prototypic autophagy receptor. So we know that oxidative stress, proteotoxic stress leads to accumulation of protein aggregates, but the same oxidative stress in a cell oxidizes P62. It triggers formation of its chains. So these oligomers are mediated by these two specific redox-sensitive cysteines in P62, and this is what allows for these chains to then pick up those aggregated proteins and degrade them via autophagy. Since we've started this, our focus was mostly on mitochondria. Conceptually, philosophically, we think that mitochondria is the center, the hub of metabolism of the cell, and also something that mediates or maintains normal health of the cell. The way how mitochondria is controlled is not just biogenesis and other sort of standard processes, but also by mitophagy. So mitophagy is a degradation of dysfunctional mitochondria by autophagy, and there's a lot of research done in the past 10 years or more. And one of these sort of the classical cascades that has been described is that protein kinase PINK1 and Parkin ubiquitin ligase are recruited to mitochondria that become damaged. So once the mitochondrium becomes damaged, and usually people use very sort of strong damaging agents to study this pathway, like depolarizing mitochondria, this leads to phosphorylation of ubiquitin, phosphorylation of Parkin, ubiquitination of the surface of mitochondria, so this mitochondria becomes ubiquitinated, and then that's when receptors And in this case, it's NDP52 primarily, but other receptors also have been implicated, which bind to it and then bring the rest of the machinery, and this autophagosome is formed and seals, engulfs this mitochondria. If it doesn't do it quickly enough, this mitochondria will simply kill the cell because it will commit apoptosis. We also know that damaged mitochondria are producing increased levels of reactive oxygen species, and Tetsushi Kataura in a lab, so he's been working to understand how this ROS signal essentially is perceived by this autophagy machinery. And the gist of the story is very simple. Well, it took a long time, but it's very simple. So this high levels of reactive oxygen species from damaged mitochondria oxidize NDP52. These chains form, and this is what makes this process very efficient. And very interestingly, it's only very efficient in humans. The other species and how these sort of processes developed in evolution is one aspect that we're quite interested in, because we see that these reactive oxygen species are only sensed by cells from humans, or somewhat basically not fully evolutionarily conserved. So, for example, these two cysteines are the ones that I just mentioned in P62 that are ROS sensitive. And we can see that in mammals here on top it's conserved, but as we go lower it starts falling apart a bit, and then in invertebrates nothing. And NDP52 is even worse case, so it's only apes and humans that they have this, essentially this mechanism. Even mice don't have this. If we look in other receptors, similar story, so that sort of blue light, light blue is something that is poor conservation. And if we compare with sort of core autophagy machinery proteins, it's very conserved. So clearly these receptors, they evolved, they've changed with evolution, and as I say, we see that these mechanisms are gradually acquiring. So it brings us to the sort of question, is it something that one of these mechanisms that allows for longevity in humans as well? And we've done some sort of reverse engineering experiments, for example, inserting human protein into mouse, for example, and given that function that exists in a human but not in a mouse. And currently with Alberto Sanz, we do this quite a sort of fanciful and perhaps not necessarily practically relevant experiment, but we try to make the mouse live longer by inserting these human mechanisms into a mouse, by giving them pink and parkin from human P62 and NDP52, with an idea that we can actually make flies do human-like mitophagy, and hopefully we'll have a fly that lives, you know, for 100 years, and then you will all hate us with Alberto, but nevertheless it'll be an ignoble price as a minimum, of course. So now, of course, this is a side issue, but we're primarily interested in what happens in humans, because ultimately whether we live beyond 10 years from now or not is another question, but we're interested in what happens in humans in terms of biology and the role of these processes in human biology. The issue is that studying this in cells, as we do, is quite difficult. As I've mentioned, in mice do not represent humans. So, for example, mouse cells are here as an example. All you need to know, the green are mitochondria. The red are supposed to be the mitochondria that are inside the lysosome. So it's a reporter specific for mitophagy. You see almost nothing happening there. If we take standard cell lines like cancer cell lines from humans, again, nothing. But then when we look at primary cells, when we take cells from somebody's skin, for example, and put the same reporter there, we see that there's suddenly loads of activity happening. So a lot of mitochondria is being degraded in normal state. So here we can actually start trying to understand why is this mitophagy going, what is the function of it. And the same is true for other types of cells. So we've tried a number of them. These are, for example, lung fibroblasts or endothelial cells. Not every cell type, which is also very important and interesting for us, for example, keratinocytes don't seem to do this very much. So there's a lot of interesting biology happening there, and clearly there's a lot of dependency on mitophagy in most of these in human cells. And so George Kelly in a lab who was for a number of years was focusing on this question. He generated this model as a result of this, a lot of studies in these types of cells, where again we see the same pattern. We see that in normal cells, without any stress, just the cells living in a dish, mitochondria produce physiologically normal levels of reactive oxygen species that are concentrated in specific mitochondria. Perhaps this reflects the damage, and this triggers that oligomerization of receptors like p62 and initiating mitophagy. What happens though in aged or senescent cells? By aged, I mean if we take cells from older people or if we senesce them in a dish, we see this very drastic change in this pathway. So reactive oxygen species physiological signaling goes down. Mitophagy is quickly declining, or simply if we take a cell from older people, mitophagy is not working anymore. And this leads to accumulation of mitochondria. So there's a massive accumulation of mitochondria with dysfunction. Recently with João Passas in Mayo Clinic, we also worked to describe the mechanism where this mitochondria also becomes partially leaky and releases DNA from mitochondria, triggering inflammatory response that contributes to inflammation that we know is associated with senescence and aging. And what's important for us here is that we think that this decline in mitophagy is the very interesting target that can restore the function of all the cells. And this is just one example of this. So here we are targeting p62 so directly. So we designed molecules that bind to p62. We screened them via high throughput screening. And so I don't want to go too much in detail, but just want to show the example of one molecule that essentially we know it binds to p62 and triggers its oligomerization, just like reactive oxygen species would do. So these are cells from young donors. This is from aged donors, so just skin biopsies. You see that these red dots are disappearing here, so mitophagy is not working in older people. And at the same time, these cells from older people are interestingly and very nicely reflecting the actual biology of human aging, which was kind of nice model for us. So they proliferate less, they increase in size, they migrate less. Normally fibroblasts need to migrate in order to do their job, deposit collagen. They migrate less, and they produce a lot less mitochondria, ATP per mitochondria. So essentially these cells are bloated, sort of sluggish, typically, metabolic function and overall function is changed. So now we treated with this compound, simply one treatment for 7 hours, and then we see that not only mitophagy is restored after the treatment, but also the cells look a lot more like cells from younger people. And the same happens in senescent cells as well. So we restore a lot of senescent markers with the treatment. So we think this decline in mitophagy is something that can be prevented and can restore the entire cascade of age-associated decline in general cellular physiology. As I say, autophagy is going down with age, and we see this especially in diseases like neurodegenerative diseases. This is a range of human brain samples from Newcastle Brain Bank that we simply blotted for p62. You can see this massive accumulation of p62 in essentially any case of or any type of neurodegenerative disease in humans, which essentially reflects the lack of autophagy turnover of components. Now, again, in tissue culture, it's difficult for us to study this because normally cells from autophagy deficient, without autophagy are absolutely fine. So they grow. This is without ATG5, the most core component of autophagy. Cells are absolutely fine. But then we ration that perhaps this is because they're not relying on mitochondria in tissue culture anymore. So we put them in galactose media as the way to enforce mitochondrial respiration, make them dependent on electron transport chain. And here we suddenly start seeing a very rapid cell death of autophagy deficiency, which is something that we see in models in vivo as well. So that allowed us to try to sort of understand the mechanism of this. And we know that this is also evolutionarily conserved. So if we lose autophagy in yeast, for example, we see a lot of cell death in the same manner. If we take cells from stem cells, human stem cells, and differentiate them into neurons, again, without autophagy, cells start dying, neurons start dying very quickly. And so this is also true for just about any way when you intervene with autophagy. So the cell will become, dead sooner or later, whether it's in vivo or in vitro. And Tetsushi, again, with Lucy Sedlakova in the lab, they did a lot of mechanistic work trying to understand why this is. So, and the usual sort of players came up. So obviously we see a lot of mitochondrial damage as a result of lack of mitochondrial turnover, increased reactive oxygen species DNA damage. But what was important was that it's the processes that's supposed to keep us alive that essentially kill these cells, because persistent activation of stress response pathways like PARPs and sirtuins, the pathways that are supposed to be dealing with DNA damage and oxidative stress, were constitutively activated, and that led to depletion of NAD in cells, simply wiping out the key energetic metabolite in the cell that essentially maintains a lot of bioenergetic reactions in the cell that led to mitochondrial depolarization and essentially cell death. So why we think it's important is because this is, first of all, very simply happens in any disease where we have mutations that lead to autophagy dysfunction. The simplest case is lysosomal storage disorders, where autophagy cascade, entirety of the cascade is perturbed. And so we've been working with Niemann-Pick disease models as well as other rare monogenic human lysosomal storage disorders models, and we see exactly the same cascade happening in all of them. So again, we see that wherever, as I say, wherever we perturb this process, and in human diseases, the same loss of mitochondrial function, depletion of NAD in a cell, and cell death occurs. And for us, this is particularly important again because we can essentially start developing interventions. So, for example, we can target downstream, so we can prevent depletion of NAD, and that rescues the cascade downstream of this whole event. But better still, we can use these models also to design new treatments. So we can use them as sort of screening platforms, for example, to screen for either existing drugs to rescue autophagy or create new drugs, and we're working with, for example, with decentralized community, Vita DAO and Molecule, to try to use these models to develop new interventions that would target autophagy and rescue, for example, defects in rare diseases, but then later also extending it to a wider range of age-related diseases. And as a sort of very sort of generic summary point is aging from all of these sort of studies, agents to me at least starts to appear as this sort of decline in metabolic activity, and because we know that a lot of metabolic cascades, everything is synchronized and orchestrated by mitochondria in a cell, and when we're young, everything is fine, so everything is ticking as normal. But then with age, we start having this mitochondria accumulating with dysfunctional properties, and essentially the entire metabolism of the cell starts declining gradually. And from where I sit, at least, actually removing this damaged mitochondria and trying to shift the balance back to the sort of youthful metabolism, there's nothing better than specifically improving the ability of autophagy to remove damaged mitochondria via mitophagy process. And this is where a lot of people working on aging essentially are also looking into, because we know that exercise and nutrition interventions, they all impact on this process and improving quality control. And obviously pharmacological interventions, hopefully we will have a better ones that will be more precise and would allow for this process to work better. So now to wrap this up, if you have any questions or would like to contact us or follow us on Twitter, for example, it's still called Twitter here, still call it Twitter. I would like to acknowledge my current and previous members of the lab and lots of our collaborators around the world. A lot of industry as well that we're working with, and there's a lot of excellent science happening outside of academia that I love to work with. And of course, this is the agents network that I'm part of, and I think, again, if you want to become part of a wider research community, please join us or any other network. So yeah, thank you.
Right, I'm biased. This is right up my street, but I thought that was fantastic. Thank you. Right, questions? Yes, Joe.
Great, very exciting. Thank you very much. I must say, I've been hoping that mitophagy really would be the falling with age, and that other people would be finding that. And you're one of the few authors who actually is enthusiastic in the same direction as we are. But how about spermidine? What about Katja Simon's spermidine story? Does that actually affect mitophagy as well as autophagy?
Well, we've never tested it. It's very interesting question. I would suspect yes, because typically everything that would affect autophagy would also activate mitophagy. I mean, I can't, I wouldn't necessarily bet my mortgage on it, but I would say that it's most likely to work. Yeah.
There's a couple of people from who've worked closely with Katja, and I'm just looking at them for clarification. Yes, you, Lauren.
I think it's not really looked at apart from cultured cells, and it doesn't decrease ROS. If you look at it with microscopic cytometry, I think it decreases overall mitochondrial load.
Yeah.
Well, it decreases mitochondrial load. That's a good start, and ROS is a complicated thing. It depends how you measure it and stuff, but it depends what you mean by ROS. ROS is not something that should be as a term even used. So typically we talk about specific production of superoxide in mitochondria or H2O2 in that area. So let's not go there. But mitochondrial load reduction is a good starting point. So it's a good indication it would work.
Yeah. And as you've said, a lot of the other things that increase autophagy also increase mitophagy. The mTOR inhibitors do that.
Absolutely.
Okay, reason?
So how do we accommodate this growing idea that autophagic processes are responsible for species lifespan differences to a sizable degree, with the evidence that calorie restriction doesn't really do much in humans?
Well, it's a good starting point to start looking into this. Interesting question. I think one of the things that we are trying to say is that autophagy and mitophagy is working better than in any other species already. So I don't know to which degree it's possible to improve it further. The speed of mitophagy in human cells compared to mouse cells is a lot higher because of these new mechanisms that are specific to humans. Whether this is anything to do with the fact that you cannot improve it any further, I can only wave hands. So I don't have an answer to that. And I also don't know why, for example, there's this any anti-aging interventions have such a different effect depending on complexity of species. Obviously, in humans, we know almost nothing. In yeast and worms, you can increase it by 100% or more, for example, the lifespan by many interventions. I'm not in a business of trying to tell you what to do with to diet or not. I'm just thinking that humans have already evolved to live much longer because everything works better than in shorter-lived species. I'm not talking about longer-lived other species which may have different mechanisms, etc.
Right. I'm really sorry, but we have time for one more question, and there was a questioner down. No, you can talk to Paul in the lunch break. There was a question here before,
right.
So sorry, I don't know.
Hi, Viktor.
Hi.
I was wondering, in your experience
Of course I do.
with your models and drugs, do you ever get overactivation of autophagy, and what would be the consequences of that?
I mean, there's definitely an idea that you can also kill the cell if you overactivate, and there are pathological states where.
So how do you define the balance there?
Well, I mean, we don't define a balance because we don't see that happening in our tissue culture models. But I can only speak for myself. So, I can sort of envisage that you can have detrimental effect, and of course, this would be down to actual practical application to any drug, for example, being formulated and titrated to the right concentration once it reaches human trials to make it safe, etc. I don't have, at this stage, we're working at a very early stage. In tissue culture, for example, we never see overactivation that would be detrimental necessarily. But I think the key here is to design, well, simply speaking, we don't have anything that would be sort of selectively activating autophagy or specific types of autophagy, full stop. So we have generic ones, but that would do all other, million other things in a cell. And for example, rapamycin is a good example of it. It will trigger just about every change in the cell that you could imagine. So, we're not there yet. We don't have so that molecule I was describing, this is the closest in my mind that we have to actually targeting specific protein that is nothing to do with signaling, and then initiate that cascade of specific mitophagy events. But that's just the first sort of example.
Okay.